Hello everyone, so I'm back with a new lecture and my name is Alina Athar and I am a lecturer in Food Science Department in RGPG College. So today's topic that I'm going to be teaching is microbiology in food. So what is microbiology? So in the context of food, it refers to the study of microorganisms such as bacteria, fungi, yeast and viruses, which in relation to food production, preservation and safety. There's like an immense importance of microbiology in the food industry, and like it helps in the role of safety and quality of food products by studying about the characteristics of different microorganisms and their impact on food spoilage and contamination as well as preservation. So let's begin with microbial spoilage. So what is spoilage? Anything which deteriorates the quality of a food product due to the activity of microorganisms. It leads to the changes in structure, taste and appearance. This is called spoilage in food, and when this spoilage is caused by microorganisms, then it's called microbial spoilage. There are like several factors which affects microbial spoilage, like temperature, humidity, pH, storage condition can also influence the rate at which microorganism can cause spoilage in food. So there are different sources of contamination. Introduction to contaminants in food. Contaminants can include microorganisms, chemicals, physical hazards that compromise the safety and quality of food. Common sources of microbial contaminations are like contamination can be due to air, water or environment, raw ingredients. So there are various sources of contamination, how the food can get contaminated. That's why it's very important to ensure that when you are eating a food item, and you have to cover it properly so that microorganisms do not contaminate it. So yeah, there are different types of microorganisms which can contaminate food and can cause food spoilage. So bacteria, fungi, viruses and so on. So before discussing more about spoilage, I would like to give emphasis on the types of microorganisms that cause food spoilage. For example, bacteria. So bacteria is one of the most important microorganism that is responsible for the contamination of food. And the characteristics of bacteria is it's a single-celled microorganism that can either be beneficial or harmful. It has like a great impact on food safety and quality. Some bacteria can spoil food through the production of enzymes or by releasing toxins. So pathogenic bacteria, when present, they can cause foodborne illness. A very important bacteria in this which causes food poisoning is Clostridium botulinum. And then we have fungi. The characteristic of fungi is it is a multi-celled organism that include molds and yeast, and it also has like a great impact on the food safety and quality. So in fungi we have molds and yeast. So molds can cause food poisoning by producing mycotoxins, and they can affect the taste and safety of food, while yeast are used in fermentation processes and it can also lead to spoilage in some cases. Then we have some other microorganisms as well, like yeast and viruses. So yeast is a single-celled microorganism which is like similar to bacteria, but it belongs to the fungus kingdom. Impact which yeast has on food safety and quality is, as I already discussed, that fermentation processes in the making or production of beverages. However, they can also spoil certain foods, especially with those if the food has like high sugar content, then high chances that yeast can spoil the food. Then viruses, they are like tiny infectious particles that requires a lot of host cell to replicate. It has a great impact on food safety and quality as well. Viruses are generally not a major concern in most food products. However, they can potentially be transmitted through contaminated water or food, which leads to foodborne illnesses. Proper hygiene and sanitation practices, they are like crucial to prevent viral contamination. So these are some of the microorganisms. Other than that, there are like a lot of microorganisms. You can see this figure of different microorganisms and how like they can cause food spoilage. And yeah, so this is like a beautiful diagram. Then let's begin with different food products and their spoilage. In particular, we have like cereals and cereal products. The impact of microorganism on grains are mold growth. Okay, mold can grow on cereals and cereal products. Then it has enzymatic activity as well. Then certain bacteria and molds, they produce enzymes that break down starches and proteins which affect the texture and nutritional quality of grains. Then rancidity, microbial activity, they can contribute to the development of rancid flavors, causing an unpleasant taste in grains. Also, it results in the loss of the nutritional value. They can metabolize nutrients in grains, which reduce the nutritional content over time in cereals and grains. They like some common cereal contaminants like Aspergillus, Fusarium species, Bacillus cereus, and Penicillium species. Penicillium is a mold that can grow on grains, and it produces mycotoxins which affect the food safety. So every contaminant has their own target. Then examples of cereal products which is susceptible to spoilage is bread, breakfast cereals, flour. So there are like a lot of examples of cereals and cereal products, and it has different organisms that can cause contamination in certain cereal items. Like bread, for bread is vulnerable to mold growth, particularly if it is stored in like humid conditions or for an extended period of time. The same way, if you talk about breakfast cereals, then high sugar content in some cereals, they can attract yeast and mold, which leads to spoilage. Then we have sugar and sugar products. You can see a very beautiful figure of sugar products. So how this product can get spoiled, there are like various microbial effects on sugars, like fermentation, microorganisms, especially yeast. It is really beneficial in fermentation process, and it can lead to the production of alcohol and carbon dioxide, which affects the stability of sugar containing products. Moreover, molds can grow on the surface of sugar products, and they can cause spoilage, visible spoilage they can cause, and potentially producing mycotoxins. So mycotoxins, they produce mycotoxins, which is like a very toxic substance. Then acid production. So what happens in sugar products? Some bacteria, they can produce acids during metabolism, and which can alter the pH of sugar containing products, and as a result it impacts the taste and stability. So there are like various contamination sources in sugar processing. You know, when the sugar is processed, at that time there's like a huge contamination due to the raw materials. You know, contaminated sugarcane or sugar beet, they can introduce microorganisms into the processing chain. And moreover, processing equipment, so it is very important to check whether the equipment is like super clean or not. You know, improperly cleaned equipment, they can harbor bacteria and microorganisms and molds, which can lead to contamination. Then air and water are like major contamination sources. Airborne and waterborne microorganisms, they can settle on sugar products during processing, packaging and storage. Then we have like major preservation techniques for sugar products, like drying, removing moisture through drying process. It helps inhibit microbial growth. Then we have packaging. Airtight packaging favors the prevention of spoilage. Like if there's the packaging is airtight, then it prevents exposure to airborne contaminants. Then we have additives. Incorporating preservatives such as sulfur dioxide they can deter microbial spoilage. Then hygiene practices, strict hygiene measures in processing facilities, they help prevent contamination. So this was all about sugar contamination. Now we are heading towards vegetables and fruits. So microbial contamination in fresh fruits and vegetables, they may be because of surface contamination. You know, some microorganisms from the soil, water or the environment, they can adhere to the surface of fruits and vegetables. Then there can be internal contamination. You know, certain pathogens can enter fruits and vegetables through wounds, pores or natural openings of the fruits, which can lead to internal contamination. That's why it's very essential to keep the fruits, it is recommended not to peel off the fruits and like keep it there for long. You know, when you have to eat fruits only, then, you know, just peel it off and then have it. Then cross contamination. During handling and processing, microorganisms, they can spread from one piece of produce to other. So cross contamination also occurs in vegetables and fruits. Then we have common spoilage organisms for vegetables and fruits, which are mold, bacteria and yeast. On fruits and vegetables, mold growth can be seen, which causes spoilage. Then bacteria like Pseudomonas and Erwinia species, they can lead to rot and decay. Then yeast can cause fermentation and spoilage in high sugar fruits, as we have already discussed about sugar products. Here, high sugar fruits, because the fruits which has like high sugar content, that's why the yeast attack on it and let the spoilage cause rest of the work. Then there are like various methods to minimize contamination, like proper washing. You know, fruits and vegetables should be properly washed, and temperature control. Cold storage inhibits the growth of spoilage microorganism. That's why it's very essential to keep the fruits in the fridge in like the proper temperature. Then sanitation practices, it's like a key to every food product, that you have to maintain sanitation practices during the processing and packaging, so that the outside contaminants cannot affect the food quality. Then genetic resistance. Developing and cultivating produce varieties with inherent resistance to common pathogens can reduce susceptibility. So these four methods are there, with the help of which you can minimize contamination in vegetables and fruits. Then we are heading towards meat and meat products. Microbial spoilage in meat can be seen in the form of bacterial action. You know, certain bacteria such as Pseudomonas and lactic acid bacteria, they can lead to spoilage by breaking down proteins and producing off flavors. There are then endogenous enzymes, you know, the enzymes present inside the meat, and they can or they can be like introduced from outside by bacteria, which can lead to the breakdown of fats and proteins, and it results in the texture changes in meat. Then we have sources of contamination in meat processing. It can be contaminated during cross-contamination, like improper handling of raw and cooked meat can lead to cross-contamination with bacteria like Salmonella and E. coli, Escherichia coli. Then contaminated equipment's not properly cleaned or sanitized, they can introduce bacteria and other microorganisms, which can lead to the spoilage in the processing chain. Contaminated raw meat entering the processing facility, they can introduce pathogens and spoilage microorganisms. So when if there are like sources of contamination in meat processing, then there are also different ways of preservation methods for meat products, like refrigeration. Maintaining low temperatures slows down microbial growth and extend the shelf life of the meat. That's why it's very necessary to keep meat and meat products inside the refrigerator. Then freezing meat at sub-zero temperatures, it inhibits microbial activity and preserves quality of the meat. Curing is a very important process. It means applying salt or a mixture of salt and nitrites, which helps to control bacteria and other microorganisms, and it enhances the flavor of cured meat products. Vacuum packaging removes oxygen, which prevents aerobic microbial growth and extends shelf life of the meat product. That was the spoilage of meat and meat products. With this, we can go towards fish and other seafoods. So there's like microbial deterioration of seafood, spoilage bacteria. Certain bacteria such as Shigella and Photobacterium species, they can contribute to the spoilage of seafood. Enzymatic activity, certain endogenous enzymes are present which can lead to the breakdown of proteins and fats in seafood. Same way it happens in meat. Yeast and mold growth, certain yeast and mold, they can cause spoilage, especially in seafood with higher fat content. Then there are like common contaminants in fish. Specifically, we are talking about fish. So there's like pathogenic bacteria, Salmonella, Vibrio, Listeria. They're examples of bacteria that can contaminate fish and cause foodborne illnesses in seafood. Then we have parasites. Some fish may harbor parasites like anisakis, which can be like a health risk if not properly handled and cooked. Heavy metals are also a source of contamination, like mercury. It can cause a health hazard in certain fish species. So there are like literally hygiene practices in seafood handling as well. Proper storage is very important. To maintain low temperature during storage, it slows down the microbial growth. Then clean processing. Strict hygiene measures during processing and packaging prevent contamination. Then we have traceability. Implementing systems to trace the source of seafood helps identify and address potential contamination issues. Last but not the least, we have cooking and pasteurization. Proper cooking and pasteurization ensure that pathogens are killed, enhancing seafood safety. Next, we are going to be like talking about eggs and poultry. So microbial risks in poultry farming, there are like two very common pathogens, bacterial pathogens, which are Salmonella and Campylobacter, which are like associated with poultry-related foodborne illnesses in human beings. Next, we have bird flu, avian influenza. It's like a viral infection that can affect both poultry and humans, emphasizing the importance of biosecurity measures. Then there are like contamination sources for egg contamination. So first of all, there's like eggshell. Okay, so outer surface contamination, in which microorganisms, they can adhere to the eggshell, and especially if it's like contaminated with fecal matter during laying. Then internal contamination means bacteria, they can enter the egg through cracks in the shell or via the egg's natural pores. So these are the two ways of egg contamination, and the prevention and control measures that we can take to control or inhibit the contamination of egg and egg products is biosecurity practices. That means implementing measures to prevent the introduction of pathogens to poultry farms. Then vaccination is a frequent practice, vaccinating poultry to prevent the spread of diseases like Salmonella. Then proper egg handling, ensuring eggs are collected properly, cleaned and stored at appropriate temperatures to minimize contamination. Then we have milk and milk products contamination, and it is basically bacterial contamination plays a major role in milk and milk product spoilage. Bacteria such as Escherichia coli and Staphylococcus aureus, they can contaminate milk during collection and processing. Then Mycobacterium avium subspecies paratuberculosis, which is also called MAP. So it's a bacterial pathogen that can affect dairy cattle and potentially contaminate milk. Then we have spoilage organisms in milk, like lactic acid bacteria. So lactic acid bacteria, it can lead to souring of milk due to the production of lactic acid. So souring of milk is a very common process, is a very common symptom that we can see in milk spoilage. And then we have psychotropic bacteria, the bacteria which is like capable of growing at refrigeration temperatures, and it also contribute to spoilage. Then there are like pasteurization and sterilization methods to prevent such contamination and spoilage in milk products. What is pasteurization? So pasteurization is basically heating milk to a specific temperature for like a set duration to kill pathogenic bacteria while preserving its quality. Then we have ultra high temperature treatment, which means subjecting milk to high temperature for a very short time to achieve sterilization, and it helps in extending the shelf life. Last but not the least, we have canned foods to be discussed in this specific topic of microbiology contamination, microbial contamination in food products, and which is canned foods. So anaerobic environment is very important. Like canned foods, they provide an anaerobic environment without oxygen. That means it enables the growth of microorganisms, aerobic spoilage microorganisms. And then heat treatment. Canning involves heat treatment to destroy or inactivate spoilage and pathogenic microorganisms. Then common spoilage organisms in canned goods are thermophilic bacteria. It's a heat-resistant bacteria that can survive the canning process and cause spoilage. Then flat sour spoilage. It's a type of spoilage which is caused by heat-resistant bacteria that produce off flavors in canned goods. Then they like importance of there's a very important way of proper sealing and sterilization in canned foods, which is like preventing contamination. To prevent contamination, properly sealed cans prevent the entry of air and microorganisms, which reduce the risk of spoilage. Ensuring safety, adequate sterilization ensures the destruction of harmful microorganisms, which enhance the safety of canned foods. So we have just completed all the specific categories of spoilage in microorganisms, the food and food products. So now it's time to discuss about detection and testing. So detection and testing is very important. Microbiological testing methods are there, like cultural methods. So in cultural methods, what we do is we utilize specific media to cultivate and identify microorganisms, which includes bacteria, yeast, fungi. And then we have molecular methods. In these methods involve techniques like PCR, polymerase chain reaction, and then to detect and identify microbial DNA or RNA. Then we have a rapid testing kit, to utilize commercially available kits for quick on-site testing for specific pathogens or indicators of microbial contamination. Last but not the least, we have plate count methods. It means it estimates the number of viable microorganisms in the sample by counting colony forming units on agar plates. So we make agar plates, and then we set the sample on the plates, and then we count colonies through specific instruments. So these are different ways to detect and test the food product is like contaminated by which pathogen. You can take measures to eradicate the food poisoning further. So detection and testing is very important. And then why we are studying microbiology of foods? It's because it has an immense importance of quality control in food industry. So what is the importance of quality control in food industry? First of all, it ensures safety. Quality control measures help identify and eliminate contaminated or adulterated food products, preventing potential harm to consumers. Then maintaining consistency. Consistent testing and quality control processes help maintain product quality, flavor, and appearance. Compliance with standards. Adhering to quality control practices ensures compliance with food safety regulations and industry standards. Then building consumer trust. So it's very important to build trust of the consumer, and for that, robust quality control builds consumer trust, delivering safe and reliable food products. So that was the importance of testing food quality in food industry. And then we have a set of regulations and standards which has to be followed. For example, FDA, US Food and Drug Administration, it enforces regulations to ensure the safety of food products in the United States. Then we have EU Food Safety Authority, which ensure food safety in European Union member countries. And then Codex Alimentarius, a very important international food standard organization, jointly run by the Food Agriculture Organization and the World Health Organization. Talking about international standards for microbial limits in food, we have ISO standards, that means International Organization for Standardization sets standards for microbial limits in various food products, ensuring global consistency. Then we have HACCP, which is Hazard Analysis and Critical Control Points. It is a systematic approach to identify, assess, and control microbial hazards in food production. So yeah, that was all about spoilage and microbiology of food. I'm going to be concluding the whole lecture by giving a summary of some key points, which is microbial control. So emphasizing the importance of controlling microorganisms throughout the food production chain to ensure product safety and quality. Then testing and quality assurance, the significance of robust microbial testing methods and quality control practices in maintaining food safety standards. So it's very important to maintain and detect, do all these testing and detection, so that safety of the food remains. And then we have compliance. Compliance means highlighting the necessity of adhering to food regulations and international standards to meet legal and ethical obligations. Now, what is the importance of microbial control in ensuring food safety? So public health protections. First of all, we have like effective microbial control safeguards public health by preventing the transmission of foodborne pathogens. If they are like good or effective microbial control, then public health will remain at like ACE. Then we have product integrity. Microbial control measures maintain the integrity of food products, which prevent spoilage and preserve quality. Then industry reputation. A commitment to microbial control enhances the reputation of the food industry, which fosters consumer trust and loyalty. So this way, we are done with the lecture, and I hope you gain insight, some knowledge of the topic related to microbiology. So microbiology is like a very important subject, and it's essential in the field of food industry as well, as we have discussed. So yeah, thank you so much for watching the whole lecture. Please do subscribe to the YouTube channel of our college and support education.